warm welcome to you guys. So once again, welcome back to your favorite and my favorite BioPoint Stream the World of Bio. Today we are having yet another super cool, super awesome and absolutely amazing lecture of cell cycle and cell division. One short session, the last minute super revision from the team BioPoint Stream the World of Bio. So welcoming you all to yet another session from BioPoint. So do hit the like button, do share it with your friends, and do not forget to subscribe to the YouTube channel BioPoint Stream the World of Bio. And if anyone new is watching this session, do not forget to join the Telegram group where we have the daily session updates, regular discussion, daily polls, and interact with me. The link for the Telegram group is t.me/need_biopoint. So, guys, welcoming you all to this auspicious platform where you can express your queries related to education on all the subjects. So, guys, welcoming you all to the Telegram group of BioPoint. And once again, reminding you, do hit the like button, share it with your friends, and subscribe YouTube channel BioPoints in the world of bio and remark us by giving a comment in the comment section. So, guys, hope you all are perfectly ready for this session. Give me thumbs up on the screen if you are ready, guys. Give me thumbs up. Okay, guys. So let's move on with this session on cell cycle and cell division. So all of you, hope you are perfectly ready for the session. So let's start with the definition of cell cycle. What is this cell cycle? It is a very important process in cells of all living organisms. That is, it is a sequence of events by which a cell duplicates its genome, synthesis other constituents of cell, and eventually divides into two daughter cells. So what are the important points? It duplicates its genome. Oh my god, what has happened? So it duplicates its genome, synthesis other constituents of cells, synthesis other cell constituents, and eventually divides into two daughter cells. This is what we say to be the cell cycle. So, and the cell cycle consists of two phases. That is an interphase consisting of a G1 phase, S phase, and G2 phase, and an M phase or a mitotic phase that consists of a karyokinesis and cytokinesis. So guys, what is this karyokinesis and cytokinesis? Karyokinesis refers to the division of nucleus and cytokinesis refers to the division of cytoplasm. So this is all about the introduction for cell cycle and cell division. Is that clear, guys? Is that clear? So, during cell division or during the division of a cell, replication of DNA and growth of the cell also takes place along with that. What all takes place? The replication. I don't know why this thickness. Replication of DNA and the other one that is growth of cell. Growth of cell also takes place. Is that clear, guys? Is that clear? 1 second. How can I change the thickness? Yeah, okay, fine. So let's move on with the next slide. Interphase. We are going to start with the topic of cell division. Interphase or resting phase. So as we all have discussed, a cell cycle consists of two main phases. That is an interphase or a resting phase. And second one is the mitotic phase or the M phase. So why this interphase is known as the resting phase? Because the interphase is known as the resting phase because the cell is not at all growing. Okay, sorry, no cell division occurs in this interphase. Therefore, it is known as resting phase. No cell division occurs. Whereas the cell is highly metabolically active in this stage. High metabolically active in this stage of interphase, but no cell division occurs. Therefore, it is known as what? Interphase. Okay, it is the phase between two successive mitotic phases or M phases. So interphase is the phase between two successive mitotic phases. Two successive mitotic phases. And in interphase, the cell grows in size and prepares itself for next division. So what is occurring in this interphase? The cell grows in size. So cell size increases, grows in size. And what happens next? The cell grows in size and prepares itself for undergoing next cell division. The next one is that it is important that the interphase lasts. The duration of this interphase is, it is important that the interphase lasts about or more than 95% of the duration of cell cycle. Of the entire cell cycle, 95% is interphase. Is that clear? Okay. So, two scientists classified this interphase into three subphases. So, who are those scientists? Those scientists are Howard and Pelk. Sorry, Pelk. So, these two scientists classified this interface into three subphases as G1 phase, G2 phase, and S phase and G2 phase. So, G1 phase, it is the first gap phase, gap 1 phase, or first gap phase, and during this stage the cell is metabolically active, grows continuously but does not replicate DNA. Does not replicate DNA. Does not replicate DNA. Okay, the cell is metabolically active but does not replicate DNA. S phase. During this S phase, what happens is the DNA synthesis occurs and the DNA content increases from 2C to 4C. That is the DNA content increases from. That is, but the number of chromosomes remains the same. That is 2. And number of chromosomes remains the same, whereas the chromosomal number, sorry, the content of the DNA increases from 2C to 4C. C refers to the content of DNA. And this G1 phase, G1 phase is the longest phase of interphase. Longest phase of interphase is the G1 phase. Then during G1 phase, most of the cell organelles increase in number. Most of the cell organelles increase in number and rapidly synthesis different types of RNA and proteins in G1 phase. Different types of RNA and proteins are synthesized in this G1 phase by G1 phase. Okay. G2 phase. Proteins are synthesized in preparation of mitosis in for mitosis while cell growth continues. While cell growth continues. This is occurring in G2 phase. Then in G2 phase, final preparations of M phase takes place. That is, special proteins and other materials required for M phase are synthesized in this G2 phase. That is, for example, if such a protein, if we name such a protein, it is what? Tubulin protein. The tubulin protein. It is synthesized in this G2 phase. Then G2 phase, and after that we have the mitotic phase. That is, it starts with the nuclear division known as karyokinesis, that is corresponding to the separation of chromosomes. Usually ends with the division of cytoplasm known as cytokinesis. Is that clear, guys? Is that clear? Then there's an intermediary stage known as quiescent stage or G0 phase. Quiescent stage or G0 phase. So as you all know, as I have taught in the previous detailed lecture, the correct order of this interface is G1 phase to G2 phase to S phase to mitotic phase to G1 phase. So while the cell is moving from this G1 phase to G2 phase, right? The cell is moving from this G1 phase to G2 phase. Okay. In the in between the pathway, there is another stage known as G0 phase. And what is the function of this G0 phase? This G0 phase checks whether this cell is suitable for undergoing the entire process of cell division, whether it can survive after that. All these are checked by this G0 phase. So some cells in adult animals do not appear to exhibit cell division. That is in case of cardiac cells or heart muscles. They do not undergo cell division, but many cells are replaced or dead instead of injured cells. These cells do not divide and further exit G1 phase to an inactive stage called quiescent stage or G0 stage of the cell cycle. In this stage, the cells remain metabolically active but no longer proliferate unless on to do so depending upon the requirement of an organism. So after moving on to the G2 phase, if it proves that the cell is capable of undergoing division, it is brought back to this G1 phase and finally moves on to G2 phase. Like that, there are certain checkpoints too. There are certain checkpoints too in order to check whether the cell is perfectly ready for this, are perfectly ready for undergoing this cell division. So first is the G1S transition checkpoint. G1 is transition checkpoint. That is the restriction point or the checkpoint. It's called as the restriction point or the checkpoint. And this is the major checkpoint. And once the cell crosses this restriction point, rest of the cell cycle is completed. If G1 phase converted to S phase, G1 phase is converted to S phase. G1 phase, the cell moves from G1 phase to X phase without by passing through this the checkpoint G1S transition. G1S transition, we can say that the cell is ready for undergoing the entire cell cycle. Another checkpoint is there between G2 and M phase, G2M transition. That is known as G2M transition. So it is a minor checkpoint. That is, cell cycle is regulated by those proteins. So the what the function of those proteins and they're perfectly working all those things, whether it will be all right after undergoing this stage. All these are checked by this G2M transition stage. Okay, so cells at this stage do not remain metabolically active but do not proliferate. Do not proliferate. Example is heart cells or cardiac cells. Is that clear? Is that clear, guys? Give me thumbs up on the screen. Give me thumbs up on the screen. I will be giving the what 10 seconds for you in order to give thumbs up on the screen. Okay, guys. Now we are moving to the next slide. That is the what a flow chart given here. Okay, a pie chart is given here, showing what exactly the cell division or the cell cycle. So let's check it. What is given here? So from the India phase, that is the G1 phase, protein synthesis and growth of nucleus. In S phase, DNA duplicates and replication of chromosomes. In G2 phase, RNA and protein synthesis. This is all what occurs in this cell cycle. Is that clear? Is that clear, guys? Okay. So in this M phase, we have the 5 different stages. That is 4 different stages. That is a prophase, metaphase, anaphase, telophase, and finally the division of cytoplasm. That is the cytokinesis. Okay, is that clear for all of you? Is this clear for all of you? Is it clear for all of you? Okay. So now we are gonna learn all this in detail. Mitosis. Prophase one. Since the number of chromosomes in the parent and the progeny of the cells is same, it is known as equational division. It is known as equational division. And the mitosis is further divided into 4 substages, that is the prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. So what happens in this prophase? What happens in this prophase? In this prophase, Replicated chromosomes each consisting of two chromatids contains and becomes visible. Contains and becomes visible. This is what happens in the prophase of mitosis. Microtubules are assembled to mitotic spindle. Microtubules are assembled to mitotic spindle. Nucleus and nuclear envelope disappear. Disappearance of nucleus and nucleolus and nuclear envelope, not nucleus, nucleolus. So centriole moves to opposite poles. All these are the important features occurring in prophase of mitosis. Metaphase. Spindle fibers attached to kinetochores of centros of chromosomes. So spindle fibers are attached to kinetochores of chromosomes, and the chromosomes align at the equatorial plane or line up at the equator of the excuse me guys, okay, equator of the spindle to form metaphase plate. This is what happens in the metaphase stage. Is that clear guys? Is that clear? Is that clear? Okay, fine. In anaphase, centromere split and chromatid separate. Centromere split and chromatid separate. Chromatid moves to opposite poles due to shortening of spindle fibers. Due to shortening of spindle fibers. Chromosomes cluster at opposite poles in telophase. Cluster at opposite poles. Nuclear envelope assembles. That is reappearance of nuclear envelope. Nucleolus, Golgi complex, and endoplasmic reticulum reforms. This is what happening in this. What to say? Telophase of telophase of mitosis. Is that clear, guys? Is that clear? Give me hearts on the screen. Give me hearts on the screen. Give me hearts on the screen right now. Right now, give me hearts on the screen. Okay, guys. Okay. So this is the figure showing mitosis, right? So here in the prophase, the disappearing nuclear membrane is shown as the centromere, disappearing nucleolus. All these are very clearly shown in this prophase. In metaphase, spindle fibers and chromosomes align at the equatorial. This is the feature of metaphase. In anaphase, contraction of spindle fibers, separation of chromatids, and their movement towards opposite poles. This is the feature of anaphase. In telophase, nuclear membrane reappearing and disappearing spindle fibers and nucleolus reappearing. This is the feature of telophase. And what is the cytokinesis? It is the process of division of the cell cytoplasm after the process of karyokinesis. That's all for that thing. And what is the difference between the cytokinesis in animals and plants? In animals and plants. Okay, so let's check it. Right. So animal cytokinesis is by the appearance of furrow in the plasma membrane, appearance of furrow in the plasma membrane, which deepens and join, dividing the cell into two. That is, it is centripetal, that is towards inside, centripetal. That is, and the other one, plant cytokinase, that is by the formation of cell plate, which moves to the, which represents the middle lamella between the walls of two adjacent cells and finally touches the cell wall and helps in its division. Is that clear? So cell plate formation or the plant cytokinesis is centrifugal in direction. Centrifugal in direction. Centrifugal in direction. Is that clear, guys? Is that clear, guys? Okay, fine. So when karyokinesis is followed by not followed by cytokinesis, a multinucleated condition arises, and this is known as syncytium. Okay. Okay, so when karyokinesis is not followed by cytokinesis, a multinucleated condition arises, and this is known as syncytium. Okay, and the significance of mitosis is the growth or addition of cells, maintenance of surface volume ratio, and maintenance of nucleocytoplasmic ratio, maintenance of chromosomal number, that is the diploid number of chromosomes, and regeneration. This is the significance of mitosis. Is that clear, guys? Is that clear? Give me thumbs up on the screen if it's perfectly clear. Fast, guys. Fast. Okay, guys. So reproduction in unicellular organisms, lower plants and some insects, repair and wound healing, vegetative reproduction in plants takes place by mitosis. So all these are the significance of mitosis. Is that clear, guys? Is that clear? Is that clear, guys? Okay, fine. So meiosis. So what is this meiosis? What is this meiosis? Comment me fast, guys. What is this meiosis? So specialized kind of cell division that reduces the number of chromosomes by half is known as meiosis. That is the reductional division. Reductional division. Okay. Occurs during gametogenesis in plants, involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2. Meiosis 1 or meiosis 2, or meiosis 1 or meiosis 2. Okay, it results in four haploid daughter cells. It results in four haploid daughter cells. Okay, interphase occurs prior to meiosis, which is similar to interphase of mitosis, except the S phase is proliferated. Except the S phase is proliferated. So, sorry, X phase is prolonged. Okay. Meiosis 1. Prophase subdivided into five phases. Prophase is subdivided. That is, prophase 1 is subdivided into five phases. That is the leptotene, zygotene, pachytene, diplotene, and diakinesis. So this is the five stages of this prophase 1 of meiosis 1. Okay. Chromosomes make their appearance as single-stranded structures and compaction of chromosome continues. This is the two features of this leptotene stage. Chromosome make their appearance as single-stranded chromosome, like the single-stranded chromosome. Okay, single-stranded chromosomes, and compaction of this chromosome continues. And in case of zygotenes, homologous chromosomes starts pairing by the process of association known as synapsis. Chromosomal synapsis is accompanied by the formation of synaptonymal complex. Complex formed by a pair of synapsed homologous chromosomes is called bivalent or tetrad. So this is all about this leptotene and zygotene. Is that clear, guys? Is that clear? Is that clear? Okay. Pachytene and diplotene. What is this pachytene and diplotene? Crossing over occurs between non-sister chromatids of homologous chromosomes. The enzymes involved in this process known as recombinase. Recombination occurs between homologous chromosomes. Recombination between homologous chromosomes is completed and exchange of genetic material occurs there. Okay. Dissolution of synaptonymal complex occurs and the recombined chromosomes separate from each other at the site of crossing over. These X-shaped structures are called as what? Chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. Diplotin can last for months or years. Okay, guys. So the X-shaped structures formed due to crossing over is known as what? Chiasmata. The figure I will show it now. Okay, so this is the figure showing all the five stages, all the six stages, including metaphase. That's why all the six stages. Okay, so look here, you can see the leptotin. In this leptotin, what you can observe is that the chromosomes form compact and like a single strand, right? And zygote the synaptonymal complex is formed. In packaging, chromosomes forms tetrad, and in diplotin, chiasmata is formed or chiasma is formed. In diakinesis, what look here, the astral rays are there moving, started moving, and finally the metaphase one five that is they are in a T equatorial plate. Is that clear, guys? Is that clear? Is that clear? How many slides left? Only three more slides left, guys. Excuse me. Okay, fine. So diakinesis, what happens is the terminalization of chiasmata. Terminalization of chiasmata occurs. Terminalization of chiasmata occurs. Chromosomes are fully condensed and meiotic spindles are assembled. Chromosomes are fully condensed and meiotic spindles are assembled. All these two things occur in diakinase that we have discussed now. These two things occur in diakinase and that we have discussed now. Metaphase. Bivalent chromosomes align at the equatorial plate. Metaphase 1. Bivalent chromosomes align at the equatorial plate. Microtubules form from opposite poles of the spindle attached to the pair of homologous chromosomes. To the pair of homologous chromosomes. Is that clear? Anaphase 1. Homologous chromosomes separate while chromatids remain associated at their centromeres. They separate while chromatid remains associated at their centromeres. Telophase 1. Nuclear membrane and nucleolus reappear. Reappearance of nucleolus and nuclear membrane. Cytokinesis follows, that is an interphase, that is a stage between two meiotic divisions that is generally short-lived, and meiosis 2 that resembles normal mitosis. And now we are going to learn, now we are learning this meiosis 1, and after that we are going to learn about this meiosis 2 that is almost same as that of mitosis. Is that clear, guys? So this is the figure showing metaphase, anaphase, and telophase. Look here in the metaphase one, you can see the chromosomes align at the equatorial plate. Okay, chromosomes align at the equatorial plate. In anaphase one, the chromosomes start moving apart from the center, that is towards the poles. In telophase one, a cleavage furrow starts deepening towards the center. This is all about this metaphase 1, anaphase 1, and telophase 1. Now we are going to learn with the prophase 2, that is metaphase 2, anaphase 2, and telophase, and significance of meiosis, soon after which session ends. The last topic of the day. The last topic of the day. That is in prophase 2, nuclear membrane disappears. Nuclear membrane disappears and the chromosomes again start becoming compact. Chromosomes again start becoming compact. In metaphase 2, the chromosomes align at the equatorial plate and they get attached to the kinetochores of sister chromatids, same as that of mitosis. In anaphase 2, splitting of the centromere of each chromosome, allowing them to move towards opposite poles of the cell, allowing them to move towards the opposite poles of the cell. That is about anaphase 2. In telophase 2, two groups of chromosomes get enclosed by nuclear envelope. The cytokinesis follows, resulting in formation of tetrad of cells. That is four haploid cells. That is four haploid cells. In significance, if we say about significance of meiosis, it is that formation of gametes, it is sexually reproducing organisms. Genetic variability, variations are very important for evolution. Maintenance of chromosomal number, that is reductional by reductional division. The normal, oh my god, the normal chromosomal number is 46 and it get reduced to 23 and 23. So that is in sperms and eggs, so that after fertilization when these few combines we get the normal chromosomal diploid number or the 2n number of 46. Is that clear, guys? Is that clear? So this is the figure showing prophase 2, metaphase 2, anaphase 2 and telophase 2. Okay, guys. So hope it's perfectly clear till now. And that's all for today. Do not forget to hit the like button. Do share it with your friends. And do not forget to subscribe to YouTube channel BioPoint. Stream the world of Bio. And once again, thank you all. Thanks a lot from the team BioPoint. Reminding you to join the Telegram group where you can interact with the teachers of BioPoint. And that's all for today. Bye bye, guys. It's me signing out from your favorite and my favorite BioPoint. Stream the world of Bio. Bye bye.